Antibody-Drug Conjugates, or ADCs, fuse the homing specificity of a monoclonal antibody with the raw lethal force of a sub-nanomolar chemical cytotoxin. Standard conventional cancer therapies lack cellular selectivity because they are highly potent small molecules that disrupt both healthy and malignant cells alike. They possess a dangerously narrow margin for safe dosing in patients. This comparison diagram maps the theoretical advantage of ADCs over those standard therapies. By tethering the payload to a targeted biologic, the drug avoids healthy tissue, effectively widening the therapeutic window. In a controlled theoretical model, this mechanism solves oncology's oldest problem. By separating targeted delivery from chemical toxicity, the industry anticipated the arrival of a true smart bomb for cancer. Based on that promise, investment surged. Between 2011 and 2021, ADCs entering early clinical trials increased tenfold. Yet, Phase 3 success and actual FDA approvals stagnated over the same period. The vast majority of these therapeutics failed late-stage clinical hurdles. The primary driver of these late-stage failures is systemic off-target toxicity. The drug attacks the exact healthy patient tissue it was engineered to avoid. An ADC functions as a four-part mechanical system. A single point of failure anywhere in the molecule collapses the therapeutic window. Achieving the ideal clinical outcome requires engineers to simultaneously balance target expression, linker stability, payload permeability, and manufacturing stoichiometry. This explodes the ADC system into its distinct physical modules: the target antigen on the cell membrane, the binding antibody, the chemical linker, and the cytotoxic payload. Target selection is the immediate point of failure. Truly exclusive tumor-only antigens are practically non-existent in human biology. Because cancer cells are derived from normal cells, healthy adjacent tissues routinely express the exact same target proteins. Regardless of how accurately the antibody binds, the payload will still be delivered to healthy organs. Roche's ADC, Kadcyla, illustrates the clinical reality of mitigating target expression overlap. This Kaplan-Meier plot shows the survival data that validated Kadcyla's approval. To salvage the drug's efficacy against standard therapies like Herceptin, researchers mandated rigorous patient pre-screening. They eliminated any trial participant who lacked exceptionally high expression of the HER2 target antigen. The clinical success of a targeted therapy often relies heavily on selectively manipulating the patient population, compensating for the biological limitations of the target itself. Moving to the payload component, a common misconception is that the most potent unconjugated toxin automatically makes the most lethal targeted drug. This compares the raw toxicity of two payload molecules. Unconjugated, the tubulin inhibitor MMAE is highly lethal. Its counterpart, MMAF, is 200 times less potent. But once both are conjugated to identical antibodies, the dynamic reverses. MMAF achieves parity or outright dominance in efficacy. Conjugating a payload to an antibody neutralizes its independent cellular access variables. It reveals that membrane permeability, rather than raw toxicity, dictates intracellular cell death. MMAF is highly hydrophilic. Once released inside the cell, it is completely trapped. It fails to penetrate the rigid lipid bilayer, allowing it to accumulate to lethal concentrations inside the targeted tumor cell. MMAE, however, possesses high chemical permeability. Once it destroys the target cell, it diffuses outward through the membrane.
This outward cellular diffusion is defined as the bystander effect. It provides a distinct therapeutic advantage. The payload eradicates neighboring tumor cells that are camouflaged or lack the target antigen entirely. But the downside is severe. Once the permeable payload crosses into the intercellular space, it loses all selectivity. It becomes a blind toxin. The exact permeability required to clear heterogeneous tumors is the primary catalyst for the systemic off-target toxicity observed in Phase 3 failures. The linker component has a singular responsibility. It must prevent the payload from detaching prematurely while navigating the reactive environment of the human bloodstream. The majority of ADCs currently failing in clinical trials share a specific chemical flaw: the instability of maleimide-derived linkers. Maleimide undergoes thiol exchange. In normal blood plasma, the linker reacts unpredictably. The bond degrades, decoupling the payload. It permanently transfers to serum albumin, a circulating protein.
The consequence is immediate. The drug becomes a free-floating toxin, circulating directly into major organ systems. Linker instability operates as a silent mechanism. It translates minor biomanufacturing chemistry flaws into uncontrolled, fatal off-target toxicity on the clinic floor. Early FDA-approved ADCs, such as Mylotarg and Kadcyla, faced an additional structural problem originating entirely in the manufacturing process. They were synthesized using stochastic lysine conjugation. Payloads were allowed to attach randomly to any of the 70 available lysine points on the antibody surface. These mass spectrometry plots visualize the result. Stochastic lysine conjugation creates a heterogeneous soup containing thousands of distinct molecular variants. This creates a wildly inconsistent drug-to-antibody ratio, or DAR. Early stochastic batches yielded molecules carrying anywhere from 0 to 10 payloads each. The low end of this ratio introduces competitive inhibitors. Antibodies with 0 attached payloads actively bind to and block cancer targets without delivering any lethal force to the tumor. The high end drives extreme toxicity. Antibodies overloaded with 8 to 10 payloads become hydrophobic, aggregating together and precipitating out of solution entirely. Without stoichiometric consistency, clinicians are dosing a complex chemical soup, making accurate pharmacokinetic prediction and reliable patient safety margins a mathematical impossibility. Modern biomanufacturing is actively engineering out the chaos of stochastic mixtures. This schematic illustrates an intermediate step used in ADCs like Adcetris and Polivy, the partial reduction of interchain cysteines. By isolating specific bonds, this reduced available conjugation sites to 8. It narrowed DAR distribution but failed to eliminate heterogeneity entirely. The modern solution now dominating late-stage clinical trials replaces random attachment with completely site-specific engineered linkers. This reaction diagram details a synthesis method using dibromomalamide, a highly stable cross-linking mechanism for drug construction. Dibromomalamide securely bridges the heavy and light antibody chains. This precise chemical lock forces the stoichiometric DAR to exactly 4 payloads on every single manufactured molecule. Synthesizing a strictly homogeneous therapeutic stabilizes the pharmacokinetic profile and widens the treatment margin. The success of targeted oncology depends on the meticulous chemical engineering of stoichiometric stability required to finally deliver on the promise of the smart bomb.